supposed to do this in 10 minutes.
Do you want to turn the podium mic off?
We did actually turn it
Cool.
off.
All right, so we know it. Okay, isolating. Thank you.
All right, there's some disclosures for me. Whatever. All right, it's April in Michigan. Not too close. It's about stink bug season, right? People who have garages and stuff, you start to see these things. And I'm going to just talk a little bit about why clinical trials stink. I thought you knew here. I was here to hear about this ICECAP trial. Well, let me just tell you why clinical trials stink first. The clinical trial is like a diagnostic test. And if you think about it, we're not just testing things that we know work, right? Because a lot of clinical trials show that what we thought worked didn't really work. It's actually like a diagnostic test, and we learn a lot about diagnostic tests in our medical training. And power is kind of finding an effective treatment when there really is one, and can be akin to sensitivity or a true positive rate. The type 1 error or alpha is sort of like 1 minus the specificity or the false positive rate. So basically, this is my analogy. Clinical trials are like diagnostic tests, and we should think of them that way. So how well are they working or functioning as diagnostic tests? And as you can see, my analogy does not stink. We make a ton of guesses when we start clinical trials, that the dose from the animal studies is close, there's no heterogeneity of effect. That's what I was talking about with Cliff before. Subgroups respond equally. We exclude some subgroups because we think that might be the best thing, even though we don't have data to say that. That we need to have some effect size to create a reasonable sample size, and that the noise in the outcomes can be understood and overcome, and that the duration of the treatment is practical. And we make a ton of compromises to reduce these parameters to make this some sort of solvable problem. But what we're really trying to explore is biology, and that there is some therapeutic response surface where we are doing a clinical trial to explore some aspect of this, that how much benefit can there be with this regimen based on these prognostic factors. And we say that when a treatment doesn't work, maybe this is a flat, or maybe there are areas where this falls under benefit, that people are harmed by treatments, and obviously that's the case. But we have to use our clinical trials to find the best regimen that provides the maximal benefit for your patient. And this is really hard. We're fitting a model on this sort of underlying biological process, and this is even this is an oversimplification. So, to switch gears, cardiac arrest is both a cardiac emergency and a neurological emergency. ICECAP is a randomized, response-adaptive, duration-finding, comparative effectiveness clinical trial with blinded outcome assessment. So that means as a researcher, I'm kind of doing everything, right? There's a little comparative effectiveness, there's a little duration finding, and also this trial does not stink. So why is there a biological reason to study therapeutic hypothermia after cardiac arrest? When this has been tested in preclinical models of cerebral ischemia, this is a summarization of 1,000 experimental treatments in acute stroke, including both focal and global ischemia models. We're counting cardiac arrest as the biggest stroke you can have. And the big green circle is, as you go up, that is how much protection is conferred by the intervention. And you go over, that's what they're ranked. So as you can see, the big green circle represents, and the size of the circles is how many studies were done. And you can see that both in focal and global ischemia models, this is one of the most well-tested preclinical treatments is hypothermia after brain injury or cerebral ischemia. And many of these studies have been reproduced. And this gives us this background as to why this is a good thing to be applying to people. And going back to that therapeutic response surface, which people should we be applying it to? People have looked at this before, and Dr. Calloway invoked a couple of the studies, the Haka and Bernard studies, looking at targeted temperature management at 33 degrees. And I'm not going to talk a lot about 33 versus 36 degrees here today, but this is a summary of many of those studies, and this is the proportion of people who are surviving with a good neurologic outcome. I look at this as encouraging, but still a lot of room for improvement. The targeted temperature management study, which compared 33 and 36, is down there on the bottom, on the second from the bottom. And FAPCA, looking at pediatrics, is there at the ultimate bottom. And interestingly enough, because of the sample size, you could see that you'd want your kid with cardiac arrest to probably be cooled to 33 based on that bar. But since the sample size was too low, the conclusion was that there was no evidence of effect. Anyway, so this is Dwight Eisenhower, and he said once, in preparing for battle, I've always found that plans are useless, but planning is indispensable. And we have spent a lot of time planning ICECAP. If ICECAP was one of my children, it would be my median child. My children ranged in age from 12 to 3. So ICECAP, if it was a boy, he'd be right between Drew and Eve. But what are we looking to prove in this study or understand? Can longer durations of hypothermia improve patient outcomes? Can the efficacy of hypothermia be confirmed by evaluating duration response? If we can show that cooling somebody longer is better than cooling somebody shorter, that's a useful answer. If we show that it doesn't matter how long you cool somebody, that's a useful answer too. And can that affect the differential safety and quality of recovery? And can we look at this both in people with shockable rhythms and non-shockable rhythms? Because the clinical trials have focused on those with shockable rhythms, because one of the assumptions that people made before was those people with non-shockable rhythms, it's not that their brain cared why their heart stopped, it's that they would have a different prognosis and it would be harder to find a treatment effect. That was why those decisions were made. So we are going to do a large randomized clinical trial, hopefully around 50 sites, 1,800 patients. And what we're going to start doing is randomizing patients to either 12, 24, or 48 hours. After we get through approximately 200 patients, we're going to fit a curve, which I'll show on a slide, which is probably easier than me just doing this, to learn whether we should be exploring different durations. Some may be longer, some may be shorter, some may be in between. And it's going to fit independent curves for both shockable and non-shockable rhythms. So the primary outcome measure that we're going to measure is the modified Rankin scale, a measure of disability. This is a functional outcome incorporating mortality, and we have our outcome assessors blinded. We use a weighting scheme to give credit to patients that have a perfect outcome, but also not transition people from being dead to severely disabled. So anyway, basically a 10 is good, a 0 is bad, which makes the sort of sense on the next scale, although here it's going from 0 to 1, but basically the mean weighted MRS is over there. And this is showing one possible biological truth that we would hope that our clinical trial could actually find. If this is what biological truth is, we are using ICECAP as that diagnostic test to say, where is the plateau? What should we be using to cool people? So this is a scenario. When you design a more complicated adaptive clinical trial, you have to do a lot of numerical simulation to see how well we'll be at answering that question. So this is a biostatistician. We don't often simulate clinical trials enough, and he feels that it's more desirable to kill computer-generated patients than real ones when calibrating design parameters. And the idea there is, if you mess up your design while the trial, and the trial is going on, that's bad. You should know what's going to happen under a variety of situations. And when we do adaptive clinical trials, we test our design under a variety of situations. So in this project, ICECAP, initially, like we said, people will be randomized in cohorts to 12, 24, and 48. Depending on what the data show, new cohorts might be opened up, or maybe they won't. If it really looks like the plateau is early, there won't be a need to open up 60 or 72. But we have to look and see what the design is doing. When we were initially planning this, the design wanted to put everybody at 6 and 72 hours. We had to say, bad design, bad design. We don't want to expose a bunch of people to those durations. And it's every microgroup, right? Like, do you think it's the self-driving cars? Could it be the self-driving cars? Self-driving cars? So basically, you have to test it by seeing what it does and then iterating it, changing the algorithm. So we built in this U-shaped sort of situation so that it would have to require additional evidence before it would start putting people in the longer durations. So we put people where they ought to be, both improving how the trial functions and improving how we learn from the results of the trial. And then the thing takes over. Oops, it took over a little too much. It's supposed to run a little movie. All right, let's see if this goes. No, that's too many back. Okay. All right, there it is. There's the movie. So patients are accruing. You're seeing how well they're doing. The open circles are what we're actually observing. The model smooths that out by saying, yeah, 60 hours looks great, but since 48 and the earlier ones don't look perfect, we're gonna smooth that out, kind of like doing a meta-analysis as they go. And then at the end, this is what it's found. And you know, when you do these simulations, how well did that work relative to the actual truth? And this was the scenario or the biologic truth that we put at the beginning. So, and this is looking at different ranges of sample sizes, how often we get the right answer. And hopefully a lot. But in the end, we hope that adaptive designs can improve discovery through clinical trials. They do require a lot more planning. And this is the culmination of a planning effort that went back to 2009, 2010, even some things before that. But our patients and our future patients need better treatments for cerebral ischemia. And we really need to bring our A game for being stewards of limited resources when we need to learn. And just so that you don't understand, so you don't believe that adaptive trials are magic, they're not always the best thing for each situation, but they can potentially allow better calibration of the scientific question and the trial conduct. They're not always smaller, they're not always better, and it can be challenging to disentangle the value of the planning process of thinking about all the things you could experience in the clinical trial from the plan itself. And the planning is indispensable, and hopefully with this plan, we'll be getting a good definitive answer on cardiac arrest for you soon. I look forward to any questions.